afternoon, everyone. I'm Natalie Piperidis, and I'm delighted to extend a warm welcome to all of you joining us today for the first SRA webinar of 2025. I would like to begin by acknowledging the Aboriginal and Torres Strait Islander peoples of this nation. We acknowledge the traditional custodian of the land on which SRA is located and where we conduct our business. We pay our respect to ancestors and elders, past and present. So today's presentation will run for about 45 minutes, and we'll have a following Q&A session. So I would like to introduce our speaker, Dr. Karen Aitken, whose presentation will unveil everything you wanted to know about the molecular markers now at our disposal in sugarcane. The title of this webinar is Genetic Tools for a Sweeter Future, Advancing Sugarcane Breeding in Australia. I feel very privileged actually today to introduce Dr. Karen Aitken. Karen is one of my mentors, in fact, and I have to say that without her, I wouldn't be here myself today. So I'm very grateful for you, Karen. I believe her to do something with the passion for what she does. And she has spent countless hours helping me through my PhD, but also through my debut in Australia. So since we have collaborated on many fundamental projects over the years, and we became very close friends. Karen has led multiple sugarcane genomic initiatives and worked in sugarcane genetics and genomics for over 25 years. She has led molecular marker development in sugarcane in Australia and has developed tools that have been utilized in the SRA sugarcane breeding program. Her interests are in identification of genes that underlie quantitative trait loci for traits that are important in the sugarcane industry, such as smut resistance, yield, and sucrose content. Karen led the development of the first sugarcane SNP array, which contained many highly informative markers for the Australian sugarcane breeding program. She also obtained a project from the US Department of Energy Joint Genome Institute to generate the first sugarcane variety genome sequence. With the support of Australian and international collaborator, the project was completed in 2024. And was published recently in Nature. So she also had like hundreds of research paper in the field. So her goal is to deliver impact from her research to the Australian sugarcane breeding program. So without further delay, Karen, the virtual floor is yours.
That didn't work.
It did work, Karen. That if you just try that again. Yeah, that's working.
Oh, right, got it. Okay, thank you very much, Natalie, for a lovely introduction. And today I'm going to talk about some of the tools that we've developed in sugarcane for use in the breeding program. Okay. So the outline of my talk is I'll start by talking a little bit about the complex genetics of sugarcane, then I'll talk about the generation of the sugarcane SNP array and the impact of the array that we've had already, and then how genetic analysis has contributed to the breeding program. So cultivated sugarcane is polyploid, it's an outcrosser, and it's highly heterozygous. So it's a hybrid between Saccharum officinarum, and officinarums are only found in Papua New Guinean gardens, and we think we're pretty sure they're a selection from Saccharum robustum with high sucrose. So this is a picture of Saccharum officinarum, and it has a phenotype that includes thick stalks with high sucrose, but not many stalks. And the hybrid is between Saccharum officinarum and Saccharum spontaneum, which is another wild type. And the origin of Saccharum spontaneum is likely to be India, but it's found throughout the tropics and subtropical regions. The reason for the initial hybrid was to increase retooning, so stalk number, and also which results in higher yield and disease resistance. But Saccharum spontaneum has a low sucrose content. So this is an example of a Saccharum spontaneum, and you can see it has many thin stalks with high fiber content. This is another accession of spontaneum, Mandalay, and that one is much more closely related or more phenotypically like Saccharum officinarum, with fewer stalks, thicker stalks, and higher sucrose. So Saccharum spontaneum is highly polymorphic. Both these species are polyploid and have different basic chromosome numbers. So Saccharum officinarum is an octoploid with a basic chromosome number of 10. So that means it has 80 chromosomes. Saccharum spontaneum is a polyploid. It has different ploidy levels, which vary from 4 to 12, and a basic chromosome number of 8. But most common is the octoploid, which gives you 64 chromosomes. But cultivated sugarcane has between 90 and 115 chromosomes. And this is a chromosome squash provided by Natalie Peperidis of a classic sugarcane cultivar. And it shows that you have, at the end of the generation of the cultivar, you have about 80% Saccharum officinarum chromosomes, which are represented by the orange chromosomes, and 10 to 15% inherited from Saccharum spontaneum, and 5 to 10% are recombinant chromosomes. So we know sugarcane is an autopolyploid, which means chromosomes randomly pair, but it has got some alloploidy in the genome that we've detected. So Saccharum spontaneum and officinarum diverged 1.5 to 2 million years ago, and the first hybrids were developed about 100 years ago. So that's fairly recent in the development of crops. Because it's only 100 years, sugarcane still has high LD, so it has large pieces of DNA. So there's not been as much recombination as there has been in other crops. What happens with the hybrids is the hybrid is produced, and then to recover the high sugar type, it's crossed back to either Saccharum officinarum or a cultivar. If it's crossed back to Saccharum officinarum, you get 2N transmission of all of the Saccharum officinarum chromosomes, and that affects the genome structure a lot in sugarcane, because you end up with a lot of regions of the genome that are completely the same as each other. So sugarcane is vegetatively propagated, and it also suffers from severe inbreeding depression. So the seed of sugarcane is heterozygous. So if you want, once you've done your selection, to keep that sugarcane cultivar, you have to vegetatively propagate it. So what are the challenges of polyploidy for genetics? Well, for the first thing you can see here is the graphical representation of the sugarcane genome, and all of the orange boxes are the Saccharum officinarum chromosomes, the yellow spontaneum, and then recombinants. So you can see that our large genome means we need a lot of data if we want to cover the whole genome. And the sugarcane genome is about 10 gigabases, and it's the most closely related diploid is sorghum, and it's highly synthenic to sorghum. So if we look at one row of chromosomes here, that represents in sorghum, which is a diploid, 1 chromosome, but in sugarcane you will have from 12 to 14 chromosomes. If you look at the different basic chromosome homology groups, you can see that there's missing chromosomes, and so it has different ploidy across the genome. So this one would be, I think, 11x, the other one is 12x. So there's variation across the genome, depending on the cultivar you're looking at. So we also, how does sugarcane work with having Saccharum officinarum as a basic chromosome number 10 and spontaneum as 8? And what we see is in the first 4 sets of chromosomes, they are completely aligned to spontaneum and pair and recombine. And the rest, the 6 other chromosomes, all 1 chromosome of spontaneum aligns back to 6 chromosomes of Saccharum officinarum. So you end up with recombinant chromosomes here, where you get crossing over, but you get interspecific recombination occurring. And you retain your spontaneum chromosomes as well. So this higher sequence similarity in functional regions hampers molecular approaches and it also increases your computational complexity of data analysis. From the point of functional complexity, you've got that potential exponential increase in gene-for-gene interactions that occurs in sugarcane because of the large number of copies of every gene. So how do we go about mapping in sugarcane? We've mostly done work using single-dose markers, and these are markers that are present in one parent as a single copy and absent in the other parent. And here you can see a diagram of you have the marker in parent 1, absent in parent 2, and in the progeny it will segregate 1-to-1. Now these markers are the most informative markers you can get in sugarcane. But they're not going to cover the whole genome, or they're going to cover most of the genome, but there will be regions that are higher dosage. So we also look at double dose markers, and they're present in 2 copies in one parent and absent in the other parent here. And you can see that these double dose markers segregate 3-to-8-to-3 in a codominant marker and 11-to-3 in a dominant marker. And then we would also be able to detect triple dose markers, and they have a ratio of 13-to-1. So by the time you get up to 3 doses, you're looking at very large populations if you want to place that marker with a high degree of confidence. Greater than 4 copies and you get no polymorphism. So we started off thinking about using markers in sugarcane over 20 years ago, and we started with amplified fragment length polymorphisms, or AFLPs, and microsatellites. We were looking for marker systems that generated a lot of markers, because if you've got over 100 chromosomes, you need to generate a fair number of markers to even have a few markers on each chromosome. So this is a classic AFLP here. And if you look at the black arrows, you can see that those are pointing to low dose or single dose markers that are segregating in this population in both the AFLP and the microsat. And the red markers show polymorphism, but it's you can see that it's not a single dose marker, so it's not segregating 1-to-1, it's segregating as a higher dose marker. If you were looking at microsatellites in a diploid, you would only see 2 bands. And so the reason we see so many bands in a microsatellite in sugarcane is that we have so many copies of that chromosome, or that particular microsatellite. So this method, we detect these running them on larger polyacrylamide gels, and it's very labor intensive and you have to manually score it. But using this method, we were able to produce up to 900 markers. So from that, we moved to capillary electrophoresis, and this is a graphical representation of what you get running your AFLPs on a automated system, capillary system. And this shows 2 different AFLPs, one with blue and one with green. And you can multiplex these. So that kind of was a really big increase. It's also very automated gel system, so it's less labor intensive. But what we found was that smaller fragments, like this fragment, this peak here, would be preferentially taken up by the capillary sequencer, and we'd end up with less markers because it wouldn't take up the bigger fragments. So we only got part of the AFLP profile. The other problem was it found it very hard to automatically score this kind of data from sugarcane, where you've got these very small peaks here that it just ignores because it sees those as not real, because it's comparing them to the other peaks, but in fact they are the single dose markers we're looking for. So we ended up having to score these manually, which took a long time, and we'd get up to 500 markers from this system. We then moved to diversity array technology, and this was microarrays, and this was automatic genotype calling. But we found that when we made the reduced representation libraries to put the fragments on the array, there was not that many low dose polymorphic markers. So a lot of the markers were high dose and didn't segregate. So we got around 300 to 600 markers from that system. But it was automatically scored, so a lot less labor intensive. So we're using these markers. We looked at genetic diversity and in the different species, and this is an example of looking at the Saccharum spontaneum accessions. So we collected 442 accessions from the world collection, and this collection is hosted we collected from a germplasm garden in America and one in Brazil. We also had 154 varieties from Australia and 101 varieties from China. And when we did the cluster analysis, you can see here that we could easily tell apart the spontaneum from the cultivars, and we could also divide the Australian varieties from the Chinese varieties. And we found when we looked at the data that 50% of the variation within the spontaneum population was captured in the breeding program already. And we think that's because there was a big integration breeding program in Australia in the 1960s. And when we looked at the data, we could see that the cultivars had a lot of Mandalay introgressed from, because that was one of the really successful crosses that happened in that integration breeding program. But the program was a lot of work and it didn't produce as much as people had hoped for, so it was stopped. When we look at just the spontaneums, we can see that they form two major groups, and group one was all of the accessions that were collected from north of the Tropic of Cancer, and group two was all of the accessions that were collected south of the Tropic of Cancer. And the spontaneum accessions clustered pretty much as you would expect from the country they were sampled from. We also identified a group 3, that was a hybrids between Saccharum spontaneum, Miscanthus and Erianthus, and then a group 4, which were not true spontaneums, but were obviously either natural hybrids that had been collected from the countries or were actually mistakes, because the germplasm gardens require a lot of looking after and they need to be replanted every 4 to 5 years. And sometimes mistakes happen. So group 4 represents spontaneums that aren't actually spontaneums but are hybrids to Saccharum officinarum, or in fact cultivars mixed up. So at this point, we realized that we needed a system that would generate a lot more polymorphic markers but would be less labor intensive. We wanted a system that could cope with the high ploidy level of sugarcane better, gave consistent results with low levels of missing data, covered the whole genome. We wanted the marker sequence because that's always useful for going back to genome sequences. We wanted to generate a large number of markers, and we wanted the format that would the genotyping data in a format that was easy to obtain with needing less bioinformatic analysis. So we chose the Affymetrix Aximon array because we knew that they had done work with strawberry, which is an octoploid, and that they had developed algorithms for work in polyploids. So they had good SNP calling ability in a polyploid background. So we knew the data was highly consistent and that large numbers of SNPs selected from known sequence that are designed to cover the entire. So we could select our own SNPs from across the genome, and we knew that there was easy and fast generation of genotyping data using the Aximon best practice workflow. And we could target the SNPs to whatever we wanted. And in this case, we wanted to generate an array that was for commercial germplasm. And this body of work was funded by Sugar Research Australia, Syngenta and CSIRO. So at that time, Syngenta had a breeding program in Brazil. So we chose 16 core lines representing the early hybrids used in the breeding programs around the world. So some of these are POJ2878 and NCO310. Those hybrids were passed around from breeding program to breeding program and were used in the background of most of the modern cultivars around the world. We also chose important varieties in Australia and some important varieties from Brazil, as well as Badilla, which is a Saccharum officinarum, so it has no spontaneum, but it's the backbone really for all of the initial hybrids that were made. So we took these 16 lines and we generated reduced representation libraries, and we generated over 3 billion 100 base pair end reads. And then we needed something to align these reads to. And fortunately, we'd already generated over 90 million paired end reads and assembled Q165, the gene-rich regions of Q165 and IJ76514. So they were assembled into 81,000 contigs, and we aligned all of our sequence from our 16 lines, 15 hybrids and 1 Saccharum officinarum to these contigs. And we selected SNPs based on there being 4 uniquely aligned reads for a single sample, 5% of all the reads in that sample supporting the variant, the quality of bases supporting the variant of at least 10, no other SNP within 20 base pairs, and biallelic SNPs only. And when we looked at the number of sites with uniquely aligned reads across the 16 lines, we found that there was, differences, but the differences in the cultivars were mostly due to the coverage we'd got from the sequencing. But one thing that stood out was that Badilla had a lot less SNPs than the rest of the lines, and that's because it doesn't contain any Saccharum spontaneum. So we took these SNPs and we calculated dosage just using a frequency of the number of reads. So we were looking for low dose SNPs, so SNPs that would be present throughout these 16 lines at a single dose or a double dose. And we calculated the frequency of the SNP by testing at different ploidy levels. So we identified from 5.9 million variants to 6 million variants. And then we filtered this by only selecting SNPs that had at least 50x coverage and ones that were calculated to be single or double dose. And we ended up with 4.1 million SNP variants at that point. So then we did a second round of filtering. And at this point, what we were looking for is not only just low dose markers, but low dose markers that actually varied in these 16 lines. So we decided to choose SNPs where the variant was present in at least 4 lines, absent in 4 lines, and present as a high dose in 1 line. So we didn't want to only select rare alleles. Then we had another filtering criteria where we looked at low dose in 4 lines, 0 dose in 4 lines, and no high dose, because we didn't want to end up with a lot of high dose SNP markers. Then we also looked at a more lenient where 2 lines were low dose, 2 were high, and there was no medium or high dose lines. And then we looked at detecting the SNPs between those 16 lines that we had sequenced, so that 60 informative crosses, to make sure that we weren't biasing one cross over another or one genotype over another. We assessed the distribution of the SNPs across the sorghum genome, and we assessed the distribution of the SNPs based on low dosage on all 16 lines, so that we hadn't biased our low dose SNPs towards some lines and not others. And we ended up with 1.83 million SNP variants at that point. And then we sent these to Affymetrix for evaluation, and they used another set of criteria for selection of SNPs, and they they wanted SNPs that had no other SNP or indel within 30 bases. They were biallelic, which is what we were after as well. SNP sequence had to be at least 71 base pairs, and they also identified regions in the contigs that were controls. They also had another set of recommendations that were based on their own parameters that could tell them something about how likely the SNP was to work in their system. So we ended up with 345,704 unique SNPs, and we put them on a large array. And we were going to filter, we decided that we'd filter this array with a population screening. And so the final SNP selection was done after we screened an association mapping population across that array, and also part of a biparental mapping population. So we could tell from the biparental mapping segregation whether they really were low dose markers or not. And we also checked the distribution across the chromosomes of sorghum as well. And we ended up with a final array of 58,028 SNPs. And they really were most of the SNPs that gave good results, so high quality SNPs from that first screening of the 345,000 SNPs, and that segregated in those populations. So even though we had gone to a lot of trouble to try and identify low dose, either single dose or double dose SNPs, we still had managed, looking at a number of reads and doing calculations, we still managed to end up with a lot of markers that were not polymorphic. So then we aligned, so when we aligned the SNPs to the R517 monoploid genome, which is very synthetic to the sorghum genome, you can see the red circle is the total number of SNPs that we had identified for the first array, which was 345,000 SNPs, and the brown circle is the SNPs that were selected for the final array. And you can see that that covers all the chromosomes. And you get this centromeric region where the frequency of the SNPs is much lower. So the results from we screened the first array with the association mapping population. So that was 480 clones was screened across the array, and we detected 48,802 SNP, polymorphic SNPs. These are the types of classes you get from this kind of analysis in sugarcane. So you end up with a number of polymorphic high resolution SNPs, and those are ones that cluster into 3 groups. So they're the ones that you would get in a diploid crop. And then in both types, all types of crops, you find the monomorphic high resolution SNPs. Then in sugarcane, we get these off-target variants that look like they are higher dose SNPs. And in some cases, like here, they can cluster them. But it's not very high quality clustering, difficult to resolve. But the one that we're mostly interested in is the no minor homozygote, and they're the ones that are single dose in the population. And then we also have the low call rate below threshold and other, which are SNPs that could not be resolved very easily into the clusters. So from this analysis, we ended up with 20,000 single dose markers and plus over 11,000 poly high resolution markers. And when we plotted the associations that we found for tons of cane, you can see here on our Manhattan plot aligned to the sorghum genome or the R570 monoploid genome that there are quite a few markers that are significant, but there's many of these, and it gave us the results we expected from something like tons of cane, because tons of cane is a highly quantitative trait, so you would expect to see lots of associations of small effect. So then we went on to look at some other traits, and this is work that was carried out by Anthony O'Connell and Jasmine Dio from Sugar Research Australia, where they were looking at identifying markers for red rot resistance in sugarcane, and they screened 305 clones, were screened across the array, and you can see, and phenotyped, and you can see here the distribution of the phenotype, so it's skewed towards resistance. And here is a resistant stem when you see no red rot, and here is a highly susceptible stem with a lot of red rot growing in it. When they did the association analysis and aligned it in a Manhattan plot to the sorghum genome, you can see there was a large effect for SMUT resistance on sorghum chromosome 5, that aligns to sorghum chromosome 5. We then went on and looked at sugarcane SMUT disease, because this disease is the most important disease of sugarcane across the world. It's caused by Sporosorium scitaminium, and it affects plants through bud or germinating shoots. And you can see here the whip-like structure of the symptoms of infection. You get those appearing. You also get stunted growth and thin weedy stalks. And you can see here some healthy cane and then some cane that is highly susceptible to SMUT. And basically you can have total crop loss if you've got some susceptible varieties. So we know from previous work that there's 2 types of resistance to SMUT. There's the external resistance, which is the bud scale or bud groove, either a physical barrier or some kind of chemical in the bud scale, and this which inhibits the growth of the spore. But there's also an internal resistance, which show classical defense resistance genes and enzymes that degrade pathogens and enzymes that generate antimicrobial compounds. So we started off by screening an association mapping population, and this was done at Woodford with at the SRA pathology farm, and we cut all of the sets to the population into 1I sets, and we inoculate them with spores, and then germinate them and incubate them for 7 days in a controlled environment, then plant them out in the field and wait 6 months, and then you can score them for presence of SMUT whips. And this takes quite a long time, about 12 months, and costs, you know, quite a lot of money. So our idea was that we could screen for markers, identify some markers, and then ultimately you could just sample the leaves, do the DNA extraction, and run the markers. It would be a lot quicker, take about 6 weeks, and be a lot cheaper. But that is very dependent on you finding markers that have larger effect. So we carried out the association analysis on 367 varieties. And when you plot that in a Manhattan plot across the sorghum genome, you find there is an effect on chromosome 5. So that's the same chromosome 5 as we saw for red rot resistance. And that is not that surprising, because since Saccharum spontaneum has been sequenced, and when we look at that chromosome, we find there is big clusters of disease resistance genes. So it looks like having those particular spontaneum chromosomes in your breeding program is going to increase your resistance to probably a number of different diseases. We also found a peak on chromosome 3, and interestingly, this peak on 5, there's also a peak on 7 as well, and that becomes important later. So we took these SNPs and we screened them across the sorghum genome and looked to see what genes we found. And we did find what we'd expected, which is resistant-like genes. So it seems like that we were finding things that we thought we would find. We then went on to test a biparental genetic mapping population. And this was a cross between Q28 and Q209. These are both important, or have been important, varieties in the Australian breeding program. Q208 is intermediate resistant, and Q209 is susceptible. So we had 320 progeny from this cross, and when we screened them across the axon array, we ended up with a total of 15,935 SNP markers that were polymorphic and we could use for genetic mapping. And we phenotype this population in 2 locations, at Woodford SRA pathology farm and in Mackay at the breeding farm there as well. So this is the genetic mapping parameters, and at the same time we decided to test genotype by sequencing, which is where you sequence fragments from all the progeny that you've got. So it's a huge amount of data that you end up with, and then you have to filter through the data to find your single dose markers. And what we found was the axon array, it produced 15,935 single dose markers across both parents, and the genotype by sequencing only produced 869 markers. And we had sequenced to a depth of greater than 20x. So really, if you wanted to get up to the axon numbers, you'd have to have a lot more sequence data. There was also a lot more missing data in the GBS data. But using both these sets of markers, we generated the maps, and we were able to assign a high amount of the linkage groups to the basic chromosome numbers. We could see that the axon map, was 3 times the size of the GBS map, which is what you'd expect because of the massive number of markers. And you could see that the axon map had a lot less small fragments than the GBS map. So we went on to place these linkage groups onto the genetic maps, and these we were lucky enough that at the same time Natalie Peperidis was generating karyotypes using whole chromosome painting to determine how many chromosomes were in each of these genotypes. And these are the graphical representations of that cytogenetic work, and that's and we were able to align our linkage groups to this. So every linkage group is assigned to one of these chromosomes, and you can see that we do cover the majority of the genome. So we went on and did QTL analysis for SMUT resistance, and you can see we found a lot of associations that were inherited from the Q208 genetic map. And when we looked at the associations that were detected in both field trials, we found there was 3 important regions of the genome on sorghum chromosome 1, which aligns to sugarcane chromosome 1, chromosome 2, and the last one was the interspecific chromosome inherited from Saccharum spontaneum. So we did find effects that were inherited from the officinarum part of the genome as well as the spontaneum part. And when you look at the associations in the map, you can see that what looks like allelic differences. So each of these has is the same gene, but it's on different chromosomes. And what we found was that we found associations that increased susceptibility as well as increased resistance. And we did the same with the Q209 map, and we found that the same regions were coming up in a lot of cases. And when we looked at the regions that again were detected in both field trials, we could see in this case 4 regions that were consistent across the two field trials. When we look more closely at the 4 top QTL we identified, you can see the biggest effect inherited from Q208 was to increase resistance, and it explained 17.4% of the variation in Mackay. And the largest effect from Q209, which is actually the susceptible variety, had an association that explained 35%, but in the Woodford and less in the Mackay field trial. So the 2 largest effects were are aligned to sugarcane chromosome 10 and 8, which is actually interspecific recombinant chromosome, so it's a spontaneum chromosome, and it increases resistance. It explains 35% of the variation in Woodford and 12% in Mackay. When we looked at the second largest effect, it was inherited from Q208, and this gene is on a different chromosome, so it's on chromosome 3, which aligns to sorghum chromosome 3, and it explains 17.4% of the variation in the Mackay data, but less in the Woodford data in this case. So those 2 experiments were done in those 2 phenotypic experiments were done in different ways. So the experiment that was running Woodford had was done as in screening normally for SMUT resistance in the breeding program. So it was dip inoculated and then planted out in the field, whereas the Mackay field trial, this was just natural infection. So we really don't know whether the differences in the results is due to different disease pressure that we would have in those different field trials, to the environment, or that it's possible that they're different isolates of SMUT, but that is very unlikely because people have screened isolates from across Australia and they are all very similar. So we think it's probably just an environmental effect. So when we looked at combining these QTL, when we combined the 2 top QTL, we found that the average SMUT incidence dropped from 65% down to about 12%. And when we combined the top 5 QTL, the disease average SMUT incidence dropped from about 79% down to less than 5%. So we are quite confident that these are markers that will work in the breeding program. We also then went on and did RNA-seq analysis on SMUT, and we had 3 genotypes, a resistant and intermediate, which was Q208. So there was the connection to the genetic analysis, and Q117. And we took samples at 5 different time points, and we did both dip inoculated and inject inoculated. So we did the dip to look at the external, but you can't tell whether you've got internal using the dip, but then we injected the spores into the bud as well to break that external resistance. So look at that. We then did the RNA sequencing, and we aligned all our reads to the 80,000 SNP chip contigs that had been used to generate the SNP array. So we were able to align all the different results from the different experiments. So from the RNA-seq, you can see that we saw many genes up and down regulated, more genes early on in the infection than later on, but and these genes changed according to time. When we did the BLAST to go analysis to look at what genes were expressed, we found that there was a lot of genes implicated in disease resistance, like leucine-rich repeats, kinases, peroxidases, and cellulose synthase. So we were quite confident that we were detecting what we expected to detect. And this work was done by Meredith McNeil at SRA. So then we went on to cross-validate the SNPs that we had identified between the mapping population and the RNA-seq, and 24 SNP markers from the RNA-seq analysis were located on the map, and we were able to see that 8 of these locations were associated with SMUC-QTL. We found that one of them was that large QTL effect that explained 38% of the variation, and was also identified in the association mapping population. So the 2 largest effects that we found in the mapping population were also detected in the association analysis population. And when we looked at the markers identified in the GWAS or association analysis, 51 SNP markers from the RNA-seq analysis were also identified in the GWAS analysis. And we found in all cases that markers were associated with increased resistance and increased susceptibility. So when we look at a schematic of what we think is going on in the interaction between the SMUC and sugarcane, we find that you get regions that cause the sugarcane to become more resistant, so cause genes to be expressed that cause the increased resistance, and then genes that are increasing susceptibility. So we realize now that we know this is a very complex interaction. It's not a single gene as other disease resistance in other diploid crops are. It has a number of interactions in sugarcane, and that's probably a lot of it is driven by the alleles that we have, the many alleles we have for each gene in sugarcane. But we did take the top markers that were identified and convert them into from the SNP array and convert them into Cas markers. And this was quite difficult. Only single dose markers can be converted. It's very hard, and you need technical validation in sugarcane, as is important as the conversion of the SNP markers to flexible high throughput marker platforms is not always possible for all SNP markers. So it's quite a complex way because you're trying to identify a single SNP in a massively high polyploid background. But we did manage to convert 2 markers, and these were the ones that were on the spontaneum chromosome that was inherited and provided the largest effect resistance. And we converted this to a CASP high throughput marker system, and it is being screened. I've only got the data up to 22, but they're screening seedlings from families and removing the ones that will probably be highly susceptible to SMUC. So we also, now that we have a lot of maps, we can start doing map comparisons. So before we had the SNP array, it's very difficult. You can't really align, AFLP markers between genetic maps. You can align microsats, but you don't usually have very many of them. So having the SNP markers has meant that we can now compare markers across the genetic maps. So the 4 maps I have here that we've done the comparison across have 7,400 markers to 9,800. With this amount of markers, you get very few unlinked markers, so you get a pretty good coverage of the map, and you can assign most of the markers to homology groups or to the basic chromosome numbers. And we found that Q208, Q209, and Q242 had very similar results in that the majority of the markers formed decent-sized linkage groups, but QC90289, which is not a cultivar but a good performing parental clone, so it's used a lot in crossing, generated a map that had a lot more fragmented linkage groups. And you can see here it had many more smaller linkage groups than the other 3 genotypes. So we were lucky enough that Natalie had also generated karyotypes for the other 2 genotypes that we're looking at, and we aligned the maps to these karyotypes. And we know that the number of chromosomes varies in these 4 karyotypes from 110 in Q208 to 112 in Q241 and QC90-289. We were able to identify the interspecific chromosomes, and we were able to see that there were 11 that we could map in Q208, Q209, and 10 in Q241, but we could only map 5 in the QC90-289, even though we know there's more there from the karyotype data. We could see that not only did the genome contain interspecific recombinant chromosomes, but it also contained translocated chromosomes. So this is where the chromosomes have recombined from groups that don't include those 6 chromosomes that are inherited from spontaneum. And we saw that each one of these cultivars had different translocated chromosomes. And we didn't find any in QC90-289, but we did find an unusual linkage group where part of chromosome 7 was inserted into the middle of chromosome 10. So we were also able to see that a lot of the chromosomes we couldn't detect. And so these were often interspecific chromosomes, as you can see here, but equally we found some saccharum officinarum like chromosomes that we couldn't find either. And so these are probably due to dosage. They're higher dose markers, and we've generated these maps with the majority of the markers are single dose markers. So because we can do map comparisons, it allows us to compare the genetic map and confirm the location of QTLs for important traits, which is important if you want to use those markers in the breeding program to select for anything. You want to know that it is the same marker that you're looking at in one genotype to another. We were able to determine that about 58% of the interspecific recombinant chromosomes inherited from Saccharum spontaneum were common between the two populations. And we think that the reason for that is there were very few initial hybrids generated 100 years ago, and then they were taken and crossed into Saccharum officinarum to recover the high sugar type, and then crossed to other cultivars. And there hasn't been that much new Saccharum spontaneum included in breeding programs. So probably those chromosomes exist as more than one copy. And when you're crossing, you're actually fixing those Saccharum spontaneums in the individuals. We lack quite a lot of markers. We still don't have complete maps, but work by Seema Vadav has used linkage disequilibrium based approaches to position unmapped SNPs, and that will be interesting to see if that gives us a lot better coverage. So for the genetics maps, despite the high number of SNP markers, so 12,000 to over 14,000 for the map, the maps are still not complete. In every map that we've generated, we've seen missing parts. All varieties are missing interspecific recombinant chromosomes, and as I said, that indicates that more than likely spontaneum chromosomes are maintained as duplicates, and that's probably the part that we detect that has preferential pairing. So within the spontaneum part of the genome, it completely randomly pairs, but it obviously does generate interspecific recombination because we see that in the karyotypes. But maybe it's more common they preferentially pair within their own species. We saw that in QC90-289 that no spontaneum interspecific chromosomes were detected for the recombinant C10-C8 chromosome. And we know from the karyotype that there are 3 of these chromosomes. So maybe they are inherited as a triplet. We really don't know at this point. But these chromosomes are not detected in the genetic maps made with single dose markers because they're obviously completely the same as each other, so they would have to be higher dose. We probably do have SNPs in there, but they don't form linkage groups because there's not enough of them. So we find that the 2N transmission of Saccharum officinarum chromosomes initially happened in the hybrids between the species means that chromosomes 1 to 4 all share a higher proportion of the SNPs in the genome than the rest of the chromosomes. These genetic maps reveal the diverse and dynamic structure of the sugarcane genome. All 3 varieties contain from 1 to 3 different interchromosomal translocations, and you would expect that to cause problems in a genome, but we think that sugarcane is able to tolerate these different translocated chromosomes because of the high ploidy in the genome. And the SNP array has been very effective, but the generation of SNPs using a different system, like an amplicon sequencing system, where we could try and determine dosage, will allow us to maybe add higher dose markers with better quality than we've been up to at the moment, and also use those dosages in genomic predictions. So the impact of the SNP array has been to generate dense genetic maps on both varieties, and we've also generated dense maps on integration clones. So you still see a large number of markers using the array in a BC1 spontaneum as well. We've identified markers associated with traits of economic importance. We've done association mapping for red rot resistance and smut resistance. We've been able to validate SNPs from RNA-seq data and from the association mapping with the genetic maps using the contig sequence. We've been able to verify the RNA-seq results through mapping back to the genetic map and the contig sequences. And we've converted the array SNP markers to the high throughput Cas markers for selection of smut-resistant families early in the breeding program. And we've been able to use the SNP sequences to identify genes that underlie trait marker associations. So the array has also started to be used for highly quantitative traits like yield and sucrose content to carry out experiments on genomic predictions. And there has been a small-scale genomic selection breeding program that started in 2020 by Wilmar International, and is running based on using the Sugarcane SNP array, and the initial results are very promising. So I wanted to just mention the genome sequence that Natalie mentioned at the start of this. It was a huge collaboration between a lot of people, but it was actually funded by the US Department of Energy and was carried out by the Joint Genome Institute. And I just wanted to point out that the data they found really backs up what we've seen with the genetic maps, in that across the sugarcane genome, the different basic chromosomes existed at different ploidy levels. And when they do the sequencing, it means that we only ever got the 67 primary chromosomes in R570, so they were the ones that we could assemble, and the rest of them were still only fragmented, so they were at different ploidy levels. So some chromosomes in the assembly seem to be shorter due to segments that have collapsed in on another chromosome. And but despite this, all haplotypes are represented in the assembly. And as Natalie said, this was published at the end of last year. So sugarcane has a complex genome structure, but the SNP array has been successfully used for genetic mapping and association studies. Both genome sequence and genetic mapping have highlighted the fact that the genome exists at different ploidy levels, that there is a great deal of architectural difference between varieties. Despite the complexity, the SNP array has enabled detection of markers that are now used early in the Australian breeding program for selection of smut-resistant families, and the sugarcane array is now being used in the breeding program for genomic predictions. We now have this first sugarcane genome sequence, which is allowing us to identify genes that underlie traits of interest. So thank you all. I just want to acknowledge all the people that have provided and worked in all these different projects that have resulted in this information. So the development of the SNP array was funded by SRA, Syngenta and CSIRO, but a lot of the work has been carried out in collaboration with SRA. So I'd like to thank Yusun and her team in Indooroopilly for a lot of the genetic marker work, Shamsul Barin and everyone at the Woodford Pathology Farm for screening the populations for smut and red rot resistance, George Preparidis and his team at Mackay for looking after our field trial there, and Felicity Aitken and her team in Moringa for the germplasm, and Xieming Wei for the association analysis. I'd also like to thank Jean Chuan Lee, Meredith McNeil and Kushla Metcalf for their contribution from CSIRO. And the genome work was a huge international collaboration, but Natalie Paparidis from Sugar Research Australia and Robert Henry and team contributed from Australia. And that's it.
Well, thank you, Karen. This was a really great presentation, and I just had like these things about back in the future when we were back in the past when we're using SNP and gels and we're fighting for it, even though we're using radioactivity. I'm glad it's a different system now. So anyway, so thanks again. So if you want to ask some question now, you can type them into the chat or raise your hand using the hand button that's located on top of the bar, and then we will unmute you, and then you will unmute yourself so you can use the microphone to ask your question when I'll prompt you. And then could you please mute again? So any question? Let me check. Okay, so far no question, but you know, I always got plenty of questions for Karen. So I'm just taking advantage that no one's asking a question. So Karen, like it seems like we need to like a lot of markers for sugarcane. Can you give like an estimation of compared to another genome, like because I mean that's been a lot of work for you and the teams. Let's say if you compare it to rice, just to have an indication, or whatever species you want, like
so
the number of marker on the SNP.
So if we compare it to sorghum, sorghum has 10 chromosomes. So if you look at putting 7,000 markers on sorghum, you would have 1,000 markers per chromosome, which is not true in sugarcane. I think nowadays people are using huge numbers of markers because often they're doing resequencing of these small genomes. So you would then have millions of SNP markers. So it's very hard to compare sugarcane to other smaller genomes because they have the advantage that they already have a lot of genome sequences, whereas we have one cultivar sequence that still isn't complete, the R570 sequence. But we probably do need to resequence because we can lines, but it's very expensive, obviously, because it's such a big genome and requires a lot of assembly. And so we don't have that advantage that other people have. But because sugarcane is so variable, like you looked at the architecture of the genome for those varieties, and you know this from your cytogenetic stuff, that they really are quite different from each other, whereas a sorghum, all their different varieties will all have the same number of chromosomes, whereas obviously in sugarcane we do not have that. So we probably are looking to generate a pan genome, which would be very useful.
Yeah, for sure. As I'm asking, what was this the time span?
For what?
I guess for developing
so this work has been carried out over 20 years. Does that make sense, or is he talking about a particular thing?
No, that was just the what was in the chat, but maybe we'll get back to that. Oh, you got a comment from George. Sorry, I have to go, but thanks, Karen, for a great presentation and sharing your works and ID. Oh, again, there's another question. So now it's in the chat. Sorry. Can't seem to find the chat. So give me a second. Nadeem, I was thinking, any genes mapping you did for sucrose contents during this project?
So yes, we did look at sucrose content. We did with the association mapping population, and we've also done crosses where we've looked at them. But sucrose has less effects than tons of cane, and we think that's because it has already been fixed in the population. So a lot of those effects have already been fixed, so that all of the association mapping population, which is all high sucrose types, all have those alleles, so they don't segregate. But we do see some effects, but they're all quite small, as you'd expect for a quantitative trait, which we kind of found disappointing because it's got higher heritability than tons of cane, but that's what we found.
interesting. So Mike Barry, do you want to ask a question?
Yeah, thank you. Thanks for that. That was really interesting. So Karen, you mentioned we're a long way short in terms of mapping the SNP markers. So where do we sit in relative terms, and what are we going to do to accelerate it? And the further question is there a cost benefit there in terms of the breeding program? Is this something we should be putting a whole heap of resource into?
I think we have the best genetic maps of anywhere at the moment. Like if you look at the published data, the maps are a lot more fragmented. I think that if you are detecting the things you want to detect, then you've got enough markers. So it's all relative to what you're doing. But if you're looking in an integration population that is incorporating new alleles, you may not be able to detect them with the SNP array. You will be able to detect SNPs if there's been recombination between the gene and the SNP you're looking at. So it really depends on what you want to do. I mean, we are covering, greater than 60% of the genome, but we still obviously have some missing regions, as you saw.
Okay, so do we need to invest more heavily is the question, or are we roughly okay?
I think it would be really good to test some of the newer methods that are coming along. Like I mentioned, amplicon sequencing. They are quite expensive, but they are. If you can amplify the right regions, then you may find, you may be able to target regions that we can't see with the single dose markers. So then you can add them into the genomic selection algorithms what you've got. So yeah, I think investing in something like that, where you could look at dosages from the reads. The only thing I'd say is we already have a lot of genotype data for the genomic selection populations, and you wouldn't want to lose that data. So you'd want to make sure you were amplifying existing SNPs as well as other random parts of the genome as well. So yeah, I do think we should invest in more technology for this.
Super good. Thanks very much.
There was a question for Dr. Varusha. Are you also working on red root disease for these? So I know you had like a slides, but maybe...
No, that project was led by Anthony and Jasmine from Sugar Research Australia. So I put it in to show that we did find a major effect, which was quite, we were all quite surprised. But it was from an international collaboration with India. So red rot disease is very important in India, and it's very variable, but it's not such an important disease in Australia. So we probably will not focus on that in the future.
Thank you for a great presentation, very informative from Jean-Yves, however. So I had another question, because you mentioned that, with the sequence in the paper, that there was a lot of resistant genes that are, on the same chromosomes. So, yeah. Because I suppose that with the SNP chip, like all the traits that are quantitative, that they're going to be very hard to, it will be it will be easier to look for genes of resistance, right?
Yes, definitely. So I think genomic selection is the way to go for the highly quantitative traits like tons of cane, and Ben Hayes, I think he talked in this series last year. He is doing a lot of work on, and his team are doing a lot of work on developing algorithms for genomic selection. So I think that's for those highly quantitative traits, that's probably the way to go. But that doesn't mean to say we shouldn't look and see if we can identify genes that underlie some of the bigger effects that we could then be able to fix them in the genomic selection analysis, and maybe that would improve the predictions. But we, like you say, yes, we would rather focus on traits like disease resistance, which, we thought that SMUT may be very simple, but when you really look at it in depth, you see that, yes, it does have a few major effects, which we've already transferred into the breeding program, but it's still quantitative. And that's what you're going to find for most things in sugarcane. I think if we were looking to integrate new disease resistance genes, we would look in spontaneum for resistance and see if we could identify germplasm that contained complete resistance to a disease and try and integrate that. And we will be able to use markers to help us do that.
Right. Yeah. There is a comment from Ananya Cartoon. Thanks for your informative presentation regarding sugarcane breeding. What do you think smut disease is most harmful compared to the other disease? In Bangladesh, red root disease is a major.
Yes, I think it's probably environmentally variable. So red rot, we have different isolates, I think, over here for red rot than they do in India. And it's maybe the environment just doesn't allow the development of that disease as much as it does in other countries. I think if the plant is stressed, it's more likely to get infected. Smut is important around the whole world, and that's why we're interested in it. It's one of the major diseases in Australia, so that's why we're more interested in working on smut than red rot.
Yeah. Hassan actually just put another comment. Appreciation for this wonderful work. What was the time span and what kind of breeding tools we can use to speed breed sugarcane, including cross-validation to varietal development?
Well, I think the SNP array is a really important tool. You can use that to look across populations if you've got different populations that you're interested in. It's a lot easier to use those SNP markers than it is to use anything else. So I think that's one of the important tools we've got. And, that is used in lots of different analysis systems, as I've said.
Another comment from Jean-Yves. Congrats for all this impressive and smart achievement in developing markers for sugarcane.
There's been a lot of people involved in that. So.
Indeed. So we got a few minutes if you have an urgent question. No. So well, thank you again, Karen, for presenting today. And I think we can all agree that we learned a lot today on the progression of the markers molecular over the last 25 years. And now we can actually use this tool really in the plant breeding program. So thank you all for your attendance today. I hope you enjoyed today's webinar and that you will join us to the next one. For information on this webinar, you can actually visit the event page on the SRA website. Thank you all. Have a great afternoon and also have a great weekend.
Thanks, Natalie.